Hello everyone and welcome back to Biology with Mrs. Evans. Today we are going to start our unit on genetics. This is probably the most interesting unit and most meaningful unit that we will cover this year, and that is because you're going to learn how you have the traits that you have, or at least some of the traits that you have. So first, let's talk about genetics. Genetics is the branch of biology that studies heredity. Heredity is basically how traits are passed down from one generation to the next, and traits basically are characteristics that are inherited. For example, you have blue eyes. That is one trait. Curly hair versus straight hair is another trait. Whether you are right-handed or left-handed, we talked about hitchhiker thumbs previously in class. All of those are traits, and then all how they're passed from one generation to the next. Those traits have been studied, and we know that some of them are dominant, some of them are recessive, and we're going to talk about what that means in a moment. So a lot of what we know about genetics came from the work of Gregor Mendel. Because of his extensive work, he is known as the father of genetics. He was an Austrian monk, and he wanted to know basically how traits were passed from one generation to the next. So he did extensive work on basically how traits were passed basically in pea plants. So there are several reasons why he chose pea plants. One of the reasons why he chose pea plants was because they have lots of traits that he could study. For an example, you see on the screen here, you have wrinkled versus round for the seed shape, the seed color. When you think of peas, you typically think of green peas, but they can be yellow. And then you see pod shape was another trait, etc. So he chose them because they had lots of traits that he could study. He also chose them because they produce lots of offspring basically in a short period of time because they reproduce quickly. And he also chose them because he could look at several different generations in a short period of time. For example, if he wanted to look at 5 generations, he could in a relatively short period of time, whereas with humans, to look at 5 generations, that's most likely not going to happen because typically we wait to 20 years or so to decide to have children. So it would be difficult to get to 5 generations. So he chose pea plants because they had lots of traits. He could end up with lots of offspring in a short period of time. He could look at lots of generations also in a short period of time. And he also chose them because he could easily manipulate them. For example, in 7th grade, you should have learned that a flower has both male and female parts. So he could simply remove the male part of a flower and control basically what flowers were able to mate with the other. So if he wanted to cross a tall plant with a short plant, he would remove the male part from both flowers, and he would take the anther, which is the male part of the flower that contains pollen, which is basically the gamete for plants, and he would dust that onto the female part, which was the pistil, and then he would control basically what flowers were able to be crossed with one another. So you'll need to know why he chose pea plants. So again, easily manipulated. He could look at lots of offspring in a short period of time, look at lots of generations, etc. So Mendel did numerous experiments looking at how traits were passed from one generation to the next. And before we go any further, let's talk about some of the shorthand that scientists used. So the P1 generation is talking about basically the parent generation, or the first crossing. And then the first set of offspring we refer to as the F1 generation. And then if you take individuals from this F1 generation and then cross them together, as you see here, we call that the F2 generation. So based on his extensive studies, Mendel came up with 3 laws, and we're going to talk about each of those in a moment. One of those was what he called the law of dominance. So as you see here, he took a short pea plant and he crossed it with a tall pea plant. And what he noticed is that when he did that, all of the offspring came out to be tall. And then when he took 2 individuals from this first generation and crossed them with each other, then he ended up with 3/4 of them being tall and 1/4 of them were short. And because of this being a consistent result that he got, he came up with a law of dominance, which we're going to talk about in a moment. So alleles. Alleles simply are different forms of a gene. And for most of the traits that we're going to talk about traits that are controlled by only 2 alleles. But later in the unit, we're also going to talk about traits that are controlled by more than 2 alleles. So for right now, we're going to look at traits that only are controlled by 2 alleles. And as scientists, we use basically shorthand to distinguish between the two. So for a dominant allele, which we're going to talk about again in a second, we use a capital letter to indicate that. And then for the recessive form of that allele, we use a lowercase letter. And when you choose the letters that you use, if you have not been given them in basically a scenario, you're going to want to choose letters that you can easily distinguish between a capital and a lowercase. So T's are good, R's are good because you can clearly distinguish between the two. H's are good. You're going to want to stay away from letters that you really can't tell the difference between a capital and a lowercase if you are not a very neat handwriter. So for example, S's are bad because if you write them similar, you really can't distinguish. C's are also bad because if you're a sloppy handwriter, you can't distinguish the two. So you're going to want to choose letters, again, that you can see a difference between a capital and a lowercase letter. So we've already talked about homologous chromosomes when we talked about basically meiosis. Remember, homologous chromosomes are chromosomes that contain the same genes on them. So here you see chromosome 4 for pea plants. In this chromosome, you will find the gene that has basically terminal flowers. And then this allele, this version of that gene, has basically axial flowers. And then in this location, you'll find the gene as to how the pod is, whether it's inflated, which is dominant, and then constricted is the recessive version of that gene, and that would be located on this area of chromosome 4. And then in this location for chromosome 4 would be basically the height of the plant. So tall is dominant, and then short, again indicated by a lowercase letter, is recessive. So for most of your genes, like I said, we're going to look at having 2 alleles. There are some that are going to have more, and we're going to talk about that later in the unit. So genes are usually described, like I said, by a capital letter if it is a dominant trait, and if it is a recessive trait, we use a lowercase letter. So some terms that you need to know are homozygous. So homozygous means having 2 identical alleles for a specific trait, and they can be homozygous dominant, such as that you see here on the screen, big T. Big T is homozygous dominant, or they can be homozygous recessive, little t. That means they have the same identical allele. So females, we're also homozygous because our genotype for our sex is XX, whereas males, they are not homozygous because they are heterozygous because they are XY. So homozygous, the other terms that you need to know that mean the same things are purebred. Purebred means that they only can give up basically one type of allele, and true breeding also means the same thing. They can only give up one type of allele because they have 2 of the same identical alleles. And then heterozygous means having 2 different alleles. So I already talked about males are example of a heterozygous individual because they have an X and a Y chromosome. And then heterozygous also means that you have a dominant and a recessive allele, as you see here. So big T, little t. So phenotype and genotype, other terms that you need to know. Phenotype is simply a physical expression of the gene, what you see. So if I talk about that I have blue eyes, I'm talking about my phenotype. My genotype is my genetic makeup. And for blue eyes, it is a recessive trait, so it would be little b would be my genotype. There are other things that impact an individual's phenotype, which we're going to talk about later in this unit. For example, environmental factors can affect how genes are going to be expressed. So Gregor Mendel's work led to him developing 3 laws, and they are the law of dominance, the law of segregation, and the law of independent assortment. And we're going to talk about each of those in a moment. So first is the law of dominance. So you heard me talk about when he crossed the tall pea plants with the short pea plant. The first generation, remember, all of them were tall, and then when he took 2 individuals from that first generation and crossed them with each other, 3/4 of them were tall and 1/4 of them were short. And he saw this time and time again, so that led him to develop the law of dominance. And what that means is that when an individual has at least one dominant allele, they're going to show the dominant trait. So an individual can be big T for, let's say, tall pea plant, so the pea plant is going to be tall. They could also be big T, little t, and they're also going to be tall because it is a dominant trait. It's always going to be expressed if present. And then the recessive trait basically is masked by a dominant trait. So the only time you're going to see a recessive trait is when the individual is homologous recessive. So that would be the short pea plant. All right, so dominant versus recessive. The second law that he came up with is basically that the alleles are going to separate independent of each other. So we know that each person gets basically a set of genes from their mom and a set of genes from them from their parent, for their dad, sorry. But you can only pass basically one of those genes to our offspring, because remember, meiosis, law of basically homologous chromosomes are going to separate, and then during meiosis 2, sister chromatids are going to separate so that each cell is haploid. So here is, let's say, a tall plant that has the genotype that you see here, big T, little t. There is a 50% chance that they are going to give the dominant allele, and there's a 50% chance that they are going to give the recessive allele to their offspring. So genes split up during meiosis and create the 50-50 chance as to how they're passed down. So law of segregation. The third law, that he came up with was the law of independent assortment. A lot of what Mendel did, as far as the traits that he studied, he was fortunate in that he chose traits that follow this simple mode of inheritance, or simple pattern of inheritance. Because of his extensive work, when we talk about traits that show the dominant recessive traits, and we see this, we refer to it as simple Mendelian genetics. And one of the things that he came up with was law of independent assortment. And what that means is that genes for different traits are inherited independently from each other. For example, you see here on the screen, you have a round pea that also has wrinkled peas, and you take that plant and you're going to cross it. There is a 25% chance that they will have basically this genetic makeup for the sex cells, because as you can see, they separate independently of each other. So the big Y can go with the big R, or it could go with the little r. So you end up with these 4 possibilities. So again, Mendel came up with this, but later, as a result of work that other scientists did, we know that there are some genes that are not going to be inherited independently of each other, and that is because they are linked together basically on the same chromosome. So for the most part, most of our traits are going to separate independently of each other, but there are a few that we now know are always going to be inherited together because they're located basically close together, on the same chromosome. So Punnett squares in 7th grade. you probably did a lot of these when you looked at doing genetic problems. And for this first part of genetic problems, you're going to be basically reviewing what you did in 7th grade. And again, what you learned in 7th grade is what we refer to as simple Mendelian genetics. There are 2 alleles for each trait, and it either was dominant or recessive. And we're going to move beyond that as we get later in this unit. So Reginald Punnett came up with a Punnett Square with being able to predict basically the offspring of parents that are crossed with one another. And that was basically probability, and everybody should know that probability is the likelihood of an outcome happening. For example, if I was to flip a coin, I would have a 50-50 chance of it being heads or tails. So 1 and 2 outcome. So for probability, again, the likelihood of something happening. So Punnett squares, we use Punnett squares to show the possible offspring from basically 2 parents mating. And you, again, should have experienced how to use these in 7th grade. So you usually write one parent on either the top, and then the other parent is on the side. And it doesn't matter if you put mom on the top, dad on the side. It doesn't matter, because the outcome is going to be the same in the squares. So just like in math class, you learned about the distributive property. You're going to basically apply that same principle to your Punnett square. So you have the genotype for, we're going to call this dad here, and this would be the genotype for mom. So you are going to distribute this little y through these 2 boxes, just like you would do in math. Same with this y is going to be distributed through these 2 boxes, and then this y is going to be distributed through these 2 boxes, and then this y is going to be distributed through these 2 boxes. And you always want to write the capital letter first, and the lowercase letter is always going to be basically in the second position. So if you cross these 2 types of pea plants, then this is the possible offspring that will be generated for those individuals. So sometimes you'll be asked to express the genotypic ratio or genotype ratio. So it would be basically 1 to 1. So 1 big Y, little y, to 1 little y, little y. And then you'll also be asked to express the phenotypic ratio, and for this one it would be 1 to 1 ratio as well. 1 yellow to 1 green. So this type of cross is what we refer to as a monohybrid cross. Monohybrid because we're only looking at 1 trait. So here is another example. We decided to cross a tall plant that is heterozygous. Remember, heterozygous means 2 opposite alleles or 2 different alleles. And then we cross another heterozygous tall plant. So notice that you have the 2 alleles here. This would be mom, this would be dad. So again, distributive property, you're going to cross, or not cross, but distribute this capital T through and distribute this little t. So if you do this, it would be big T. And then now you're going to distribute this T through. So if you were to ask, or if you were asked to tell what's the chance that this crossing could create an individual that is short, you would say that it is 25%, because 1 out of 4 should be short, and then 3 out of 4 will be tall. And that is, again, the phenotypic ratio, whereas the genotypic ratio for this one would be 1 big T to 2 big T little t to 1 little t, or simply we usually just write it as a 1 to 2 to 1 ratio. So dihybrid crosses are crosses where you want to see what's the likelihood of producing offspring that have 2 of the traits. So notice up here at the top, law of independent assortment, and that you are crossing basically 2 individuals that are heterozygous. So that means they are big R, right, and they are crossed with each other, so the other one would be the same. So law of independent assortment, remember, we're going to show basically the 4 possibilities for both parents. So this would be, let's say, mom, and then this would be dad. So here are the 4 possibilities. They could be big R, big Y, big R, little y, little r, little y, or little r, little y. And the same is true for the dad, because we chose 2 individuals that were heterozygous. So you notice the possibility for the offspring. And again, this would be a dihybrid cross. You could do the same thing for 3 traits, and we call that a trihybrid cross. For this class, you need to be able to do, as far as a dihybrid cross would be, usually I will set it up, or the people in the SOL will set it up for you like this, and you just simply have to use basically the distributive property like we just did to come up with what would be the inside. Extra credit, you will get a extra credit question from me that you will actually have to set it up yourselves, but for the most part, like I said, you'll get it in this form, and then you have to figure out what the sides are, or insides, based on using the distributive property. So probability, again, we use Punnett squares to figure out the probability of potential offspring from a particular crossing. So that's it for this first part, basically, which is simple genetics, and we're going to talk about what happens when traits don't follow this simple pattern of inheritance. So that's it. Hope you have a great day. Talk to you later. Bye.